Hello, everyone. Welcome to today's live broadcast, Nitrosamine Risk Reduction in Pharmaceutical Formulations, a collaborative approach between industry and excipient providers. I'm Terry Summers, the Senior Editor for Pharmaceutical Technology, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by IFF. I'd like to share a statement from our sponsor before we get started. IFF's Pharma Solutions experts have decades of experience in the pharmaceutical industry. They provide customization and innovation support and differentiate you in your market, and can provide support by working in collaboration across the complete drug development pipeline. IFF Pharma Solutions pioneered excipients like Avicel and Methacel nearly 70 years ago, and has since created a unique range of well-known excipients designed to deliver performance and cost advantages across oral dosage, oral liquids, and topicals. Now, as part of IFF, Pharma Solutions is an even stronger partner in navigating global regulatory and supply chain complexities, delivering what is best for you. A commitment to quality, service, and reliability isn't to check a box, but to drive an endless pursuit of delivering the right answers exactly when you need them. You can learn more at pharma.iff.com. And we have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge a slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I'd now like to introduce today's speakers. We're pleased to be joined today by Justin Moser, Principal Scientist, Vaccine Drug Product Development at Merck, and Meinhof Brockhagen, Principal Pharma Application Specialist at IFF. Justin has a BS in chemical engineering from the Pennsylvania State University. He's working in vaccine drug product development, covering the exit of discovery through phase 3, commercialization, and launch. His prior focus was on leading Merck's cross-functional and Nitrosamine's core risk team, which authored and submitted the risk assessment outcomes to health authorities with a personal focus and interest in addressing risks in drug product formulation and processing. Dr. Brockhagen's responsibilities at IFF include technical service and development for the pharma application markets, as well as in-depth analysis of structure property relationships used to commercialize new products and assist customers' developments. Dr. Brockhagen holds a diploma in organic chemistry from the Technical University of Dortmund, Germany, and a PhD in carbohydrate chemistry from the University of Rostock, also in Germany. He is a certified Six Sigma Black Belt. He's the author or co-author of more than 40 published patent applications and 8 articles in peer-reviewed journals. Thank you both for joining us today. Justin, please get us started.
Thank you very much, Terry. And pleased to be here today to talk about this subject. Really appreciate the invite, and hopefully the audience gets some useful information out of today's discussion. So what I'm going to talk to you about is introducing the N-nitrosamine risk problem for the pharmaceutical industry, and how the industry and health authorities have worked together to assess and deal with these risks in our drug products up until this point. And that's through use of structured risk assessments collaboration, and also development of formation models to guide risk assessment. An outline for today's discussion. First, I'll give a brief overview of the challenge that the industry faced initially, and hopefully and you'll see how that progressed up until this point. Discuss the common risk factors that we've identified for nitrosamine formation, or abbreviated as NNO, in both drug substance and drug product. Discuss how we've developed and used a model amine studies to confirm which risk factors are prioritized and important and which ones are not. And then also how to use those models to guide assessment of your own drug product risks for nitrosamine presence and formation. And then outline at least an example or two of development of predictive models that can be used in the appropriate setting with the appropriate informed background information on your product to assess risk and quantify risk to finalize your risk assessment, and then discuss some areas of research that remain for next level understanding and control. So what is the problem that the industry has been dealing with? N-nitrosamines are not uncommon. The chemistry has been well known. In fact, it's been used as precursors in drug substance or active pharmaceutical ingredient synthesis for many years in a controlled manner. But what was discovered, and this came about in 2018 with the discovery of NDMA or N-dimethylamine, N-nitrosodimethylamine in valsartan. Following very shortly after the discovery of these impurities in metformin and other drug products. This prompted the health authorities to react and ask that all drug product manufacturers and market authorization holders assess the risk of nitrosamines in each and every drug product. As you can imagine, for many companies, this could be quite a lot of drug products in their commercial portfolio. And then shortly thereafter, the attention turned to development pipeline for new products coming forward. And at the time, there was very little knowledge of N-nitrosamine formation risk or presence in drug products. We had literature about the chemistries at play, and also some of the literature about the types of reagents like the nitrosating agents that could be present that can react with secondary amines to form a nitrosamine. And I want to highlight that this is a chemistry up at the top that we're dealing with. You have a secondary group to form the nitrosamine and kick out the hydrogen atom. There is various chemistry that can lead to this secondary amine. It can form from a tertiary amine, quaternary amine, and or tertiary amide. So there's various routes that you can get to this reagent. And then also for the NO group, there's various of nitrosating agents that can deliver this NO group to attack that secondary amine and form the nitrosamine. So very quickly, the breadth of this problem was very apparent. The literature and knowledge available to help guide formulation and product development scientists and industry is very limited. So we had to start digging. And upon that digging, one of the things that was very disheartening and that Meinhof will elaborate on in quite depth is that we started to find publications and also generate data to show that excipients, pretty much most excipients that we use in drug product formulations can carry ppm levels, give or take, of nitrite impurities in them. And the nitrite in the presence of water can form nitrous acid at certain pH and then go on to form these different nitrosating agents. So this made this apparent, this problem very apparent and potentially real in specific drug products and required attention. So what approach did the industry and health authorities work together to deal with this problem? First, there's a lot of knowledge that we've leveraged and continue to leverage that's existing out there for pharmaceutical companies to use. Several ICH guidelines on impurity control, addressing and constructing risk assessments like Q9, and then also M7 on how to deal with genotoxic carcinogenic impurities. Also, very nitrosamine specific guidances started to come out, particularly one very early on that's still very strong and in use today is the Article 5.3 from EMA. Very important guidance on how we can address this risk. And then industrial consortia were formed that shared knowledge and are still in existence today, like the Innovation and Quality Council, or IQ, EFPIA, Pharma, and other industrial consortia. I certainly encourage folks to get involved in a lot of very robust information. A lot of the publications I will cite today were done through collaboration of scientists dealing with this problem within that consortia. And then also industrial collaborations like I cite here with the one with LASA, which LASA led the construction of a blinded database of data supplied by, over, I think, 20 companies across the industry of excipient characterization data for nitrite impurities. Then from this, we, really that information helps build and strengthen the information gathering and scientific foundation. And also with the conjunction of literature on nitrosamine reactions, It helps define what are the known risk factors, what pH range is really important, what types of concentrations are important, and what types of amines are more reactive than others. And then we already had a strong level of information available on our products. Dossiers and specifications form constraints for how you can constrain your risk assessment and what types of values for reagents or process conditions or storage conditions are relevant based on the control strategies in place. Questionnaires to the excipient manufacturers and also manufacturers of drug products gathered a lot of relevant information. And when that wasn't enough to help de-risk the product, then you needed to go to experimentation. And there is where a lot of model amine studies I'll capture really build a lot of good data and knowledge. Nitrite impurity characterization I mentioned, and then also reactivity experiments. And through this, in published literature, we're able to build some quantitative models that can be used to estimate your risk in a quantitative fashion. And then finally, if you do have some potentially forming nitrosamine in your product, and you have some estimates of worst case formation, what is the potential acceptable limits that you could have in your product? And there's evolving even today. There's still guidance evolving on how to define that, but acceptable daily intakes can be estimated, and that process is outlined in M7, and that can give you your goal line, if you will, for what's an acceptable daily intake for your product. And then based on your estimates, are you even close to that where you need to say this is a risk and I must proceed to testing? Or is the estimates orders of magnitude below that, for example, where you can say this is a negligible risk to the patient because of all the information and assessment you've built to date? So what types of factors are important for assessing risk in a drug product or drug substance? On the left, first is your potential reactants for formation. You have your source of a vulnerable amine. This could come in the API structure. Secondary amine modalities can be present in the structure itself, and that's where you get to your NDSRIs. You could have impurities that were used in the synthesis of the API. A degradate formation is another route that needs to be considered. That could be outlined in your dossier or development reports. And then also potentially in certain excipients, you can have sources that can of reagents that can lead to a secondary amine. Properties of the vulnerable amine listed there, and I'll get into some of that a little bit further in some of the model amine studies that are important. Also, the source and concentration of nitrosating agents. Nitrite impurities and excipients, I mentioned that they're at the ppm level, but we're trying to control nitrosamines generally in the ppb level. So ppm 3 orders of magnitude higher than what we're trying to control at is actually a pretty relevant concentration. So this is where the relevance of this discussion today and the work that excipient manufacturers are doing very important. On the process side, drug substance, you consider the proximity. If you have an amine and a nitrosating agent, are they going to be in the same reactor, same tank? Or are they separated by several steps and also several purification steps where you can apply purge factor calculations to estimate and help inform your risk? Time, temperature, and pH control in your process can also be important. Drug product formulation. This is not an extensive list for any of these factors, just some of the highlights. The physical form of the reactants, I'll highlight that some data later, is an important piece. Certainly the dosage form, if it's a liquid or solid, makes a big difference in the risk. Liquid and being much higher risk due to the mobility of the reagents to see themselves and react. Shelf life conditions need to be considered, and moisture levels in your product. We've shown in published literature, multiple publications that show that can be very important. And then the process, what types of unit operations do you have? Is there a presence of water? I think the thing that we haven't done enough of is a scale of mixing uniformity. We know in solid dosage formulations and other types of research that this is not a perfectly mixed system. You could have pockets of enriched drug or other components. And then if that's the problem child and you have a enrichment of the reactants, you don't, and we're dealing with the ppb level, you don't need much to form an unacceptable level in a solid dosage form. So mixing uniformity and assessing that can be important. I'm going to skip over this because Meinhoff's going to cover the excipients in good detail. So here's an example, and there's a publication cited there with a lot more information. That's actually, there's two, I just want to put a pitch out there, two very important special editions, one published in Journal of Pharm Sci, and the other one recently published in Organic Process Research and Development, that have a lot of good publications on this topic. So I encourage everyone that want to read more and learn more to look those up. This was work that's published looking at several different types of model amines, different structures, different physical forms in terms of being a salt or neutral form, also different pKa's. And the work here that was done was to formulate these in different formulations with different types of excipients in a controlled manner, using dry and wet processes, looking at blends versus compressed tablets, and assessing the different levels of formation risk that we see, or different levels of nitrosamine that form that would quantify to the level of risk present. And first thing that was discovered early on in this research is that this was not expected by people in the industry, but we learned very quickly that even in a dry, direct compressed tablet, depending on the conditions and the amount of amine and nitrosating agent present, or nitrites in your excipients, you can form high levels of nitrosamine in those type of systems. This particular data set captures several things. One is the influence of storage humidity on the amount of nitrosamine you can form. The other thing that we detected upon further investigation is that amine 1 here actually lost its crystallinity when we got up to 75% RH. So when you lose your crystallinity and turn amorphous, you can have more effective mobility in your formulation and also surface area for reaction. And those are things we know about amorphous materials in general versus their crystalline counterparts. And we speculate that that had a contribution to the elevated levels of nitrosamine that we saw in these types of cases. And also in that publication, we showed the importance of pKa of the vulnerable amine and how that can drive things. We showed that even at 10 ppm level of nitrites, purposely controlled in the formulation, you can get formation of levels of nitrosamine that can be concerning, and then other factors that were explored in that work. Another example I wanted to highlight was a publication here by several of my colleagues in the IQ group on the development of a quantitative model of nitrosamine formation in aqueous medias. And this is for dialkyl amines. So not appropriate for complex or cyclic amines, but for dialkyl amines, this model was developed to be conservative and quantitative in its estimates. And what this group did was they surveyed the literature. They found the different equilibrium reactions present to form, the nitrous acid that leads to nitrosamine or nitrosating agent formation, as well as the protonated or non-protonated state of your secondary amine. And then the three main driving equations that lead to nitrosamine formation through the dinitrogen trioxide route, through the dinitrous dihydrogen route, and then also through nitrosyl halides, which can form from the presence of things like hydrochloric salts or other things that you can get this dinitrile route here. And then also, the authors purposely designed this model to be conservative. And two things that really kind of call out that they did was, first is they surveyed the literature for all the different reported rate constants and used the highest ones they found to drive the most nitrosamine formation. The other thing that's very well published and known is nitrous acid will degrade in its own right. And that is the source of all the nitrosating agents, and that was ignored in the model. So by ignoring that, you're going to keep a higher concentration of nitrous acid around to form the nitrosating agents and drive the nitrosamine formation. So this was done very intentionally. So when a user would use the model to predict nitrosamine formation, they can be confident that with the right input information from the product and conservative values that they enter, the model is conservatively predicting that if the result is still below your acceptable daily intake or known limit of control for the nitrosamine, you can be confident that you're predicting a no-risk scenario in your product. We went on to further support this model by doing some experimental studies in aqueous solution using this 1,4-phenylpiperidine HCl salt, which was amine 1 from the previous work in the solid dosage monoamine study, and looked at it at two different concentrations of nitrite, one which was around 0.1 millimolar, or a pretty high concentration in solution for nitrite, and the other one which was at 10 to the minus 4 millimolar, this data here is at 0.1 molar, and then we did one at 0.1 millimolar. And this was estimated to be a kind of a typical order of magnitude of nitrite that you might have in a liquid drug product formulation. And what we showed in those two experiments, they were done at different pHs, is that in fact the model here in the solid line is over predicting the amount of nitrosamine or level extent of nitrosation that you would expect compared to the experimental data. And also it verified, as we would expect, that as you moved away from the pKa of nitrous acid, which is around 3, to higher pH, your rate of formation goes down to negligible amounts and insignificant amounts. So the model is successful at proving that against experimental data and part of a verification. So certainly a good tool for those to have when they're assessing their risk in drug product. And this is just an example of how then once you had that model constructed and that was solved using a Berkeley-Madonna type methodology, you can set this up in a software like Dynachem or potentially MATLAB to solve those equations simultaneously, and then build a common user interface. In this example was done in Excel, so that users can go and put their amine information, their dose information, what their formulation is, either known nitrite concentrations or assumed nitrite concentrations that are conservative. And with that formulation information, it can calculate in a dosage form how much nitrite's present, the pH, and time. So is this are you doing a simulation on a process step that might be 10 hours, or are you trying to simulate a liquid drug product that has a 2-year storage, or therefore 730 days roughly as a shelf life? And just to give an example of how one might do this appropriately to make sure their estimate's conservative, you look up in your dossier and you find out that you look up your data and you find out that the mean has a pKa of about 10.5 plus or minus 3, known variability, so you use a conservative estimate of a lower pKa. You also find out information about your excipients, and if you don't have particular nitrite information on the excipients you're using, you can use a conservative estimate like 5 ppm, which is much higher than what the data suggests would be a conservative estimate, like based on this publication down here from the LASA project. And then you can look at your pH range and control in your process or final product and use something that's lower, that's going to drive more of that nitrous acid-based nitrosation. And then you simulate and find out that you have about 0.5 nanograms of nitrosamine potentially expected over the shelf life. And that's much less than the 18 nanograms per day, a generic limit set for an NDSRI based on the available guidances. So then you can suggest to yourself and confirm that based on all the information, which is comprehensive, and this model, which is conservatively designed, we're much below any kind of concerning risk on a daily dose for this product, and conclude potentially that you have no risk of nitrosamine formation and exposure above the daily intake of your product at hand. The last thing I want to discuss is also there's work that's been done to assess blister packaging process analysis. This came about some work published out of the Novartis company, and then also several researchers that investigated further that you can have nitrosamine formation risk in blister packaging. You have nitrocellulose can decompose and form nitric oxide that is a direct nitrosating agent. And also we found out that the inks can have amines such as diethylamine and dimethylamine present in the inks. And you can have direct reaction in the inks or overlacquers to form nitrosamines, which has been detected in the foil provided from the suppliers. And also upon application of heat in the blister process, you could further enhance that reaction and then even volatilize the nitrosamines that could potentially contaminate the open product in the line. And I encourage everyone to go to this publication to learn a lot more. There were some experiments done where some of these coupons of this blister foil were cut to be representative of surface area that might be expected for a common 500 milligram tablet formulation, for example. These were sealed in a vapor impervious vial and heated in a lighthouse chamber. And using this nitrogen phosphorus detection, the nitrosamine, NDMA and NDEA, was tracked over time, the amount released over time in this heated experiment to try to simulate time temperature that's the foil would be experiencing on the blister line. So we can get a rate of formation for these different nitrosamines. And NDMA is not surprisingly at a higher rate. It's a more volatile and a higher vapor pressure than NDEA. And then coupling this with assessment of the blister line. So for aluminum foil packaging, once the foil and the open cavities go under the sealing line, whether it's a roller or a press, it seals that aluminum foil directly on the aluminum cavity below it, and there's no direct contact of the inks or overlacquer to the product. Therefore, the only practical route for contamination would be volatilization of nitrosamines or amines into the air, upstream in the blister line, and then deposit or capture in the open blister wells that are being filled with your product. So it's a pretty low probability that that's going to happen to any common extent. And you can also find out that in your clean room, you typically have air exchanges going on at a certain rate. So that's going to help remove any nitrosamine that's formed as a function of time based on the rate of air exchange in your room. Then you can form a model, a process model using this rate of formation and a rate of removal of nitrogen or nitrosamine that could be present based on air exchanges, and look for when you hit steady state to see how much nitrosamine may end up in a certain volume of blister well, and assuming all of it absorbs into your product, what the maximum concentration would be due to the blister packaging. And in this case, using conservative assumptions of a typical blister line, we found the risk to be quite negligible based on this type of model that we built. Finally, some conclusions. Industry and health authority have worked tirelessly to kind of help build structural approaches on how to assess risk. And a lot of the data and learnings from some of the experimental studies I highlighted today have been used to articulate risk and considerations in these workflows, such as the one that's published by EFP on N-nitrosamine risk assessment workflow. Secondary amines and concerning nitrosable substances can be present in APIs. There's a wealth of data to show nitrite impurities are typically present in excipients, most of them, at varying levels. And also, therefore, in some drug products where you have these two reactants or reagents present, you have a theoretical risk of formation. However, unless you want to send every product to secondary testing and confirmation, which is unnecessary for many, there's a lot of published work, experimental and models being developed that I shared with you today that can be used to help quantify risk based on the input knowledge of your product and process control strategies for those products to conservatively estimate formation. And then using the established practices ICH-M7 or known information on those nitrosamines to set limits, daily limits, you can compare your quantification against those limits to say whether or not you have a risk or not. Based on the assessment and published information, that blister example I gave, really that work has shown negligible risk of nitrosamine contamination directly from aluminum foil packaging in well-ventilated clean rooms. If you have a sealed operation, that would be a different assessment, but in well-ventilated clean rooms, that work shows negligible risk is present. I think this work that has been done today really has provided in a short period of time, only a few years, some good foundation that can help us understand risk in drug products. I want to also highlight, in certain cases, if the deeper understanding is needed, particularly when you're developing new products and you're thinking about the science and mechanisms going on, I want to highlight a few things to consider. One is, don't overlook the physical form of both of your reagents, both the nitrosating agent and the amine. I think I showed data clearly showing that a physical form of the one amine can matter very greatly. The other thing is, at ppm levels of nitrite and excipients, we've seen that that can be relevant in forming concerning levels of nitrosamines. To me, that speaks to, this is very supported by surface chemistry type reactions that are almost certainly at play, particularly because the presence of moisture interstitial within solid dosage products have shown this in stability studies. Wet granulation and application of water to surface these particles have shown elevated level of risk. So understanding that chemistry better and where those reactions are occurring could lead to deeper understanding of what's going on scientifically, to lead to more predictive and models, and get away from conservative estimates, which today are very, very conservative and tend to incredibly over-predict what might actually be in the product. And a few acknowledgments. Just wanted to acknowledge members of the nitrosamine team at Merck, numerous discussions within the IQ nitrosamine team, authors of the various cited publications, which I had a lot of work side by side with them in their dedicated efforts to put this science out there. And also just wanted to thank the organizers from Pharmaceutical Technology and IFF for inviting me to speak today and putting on this webinar. And hopefully everybody's found the information very useful. So at this time, I'd like to turn it over to Meinhof Brockhagen to speak further about what's going on with excipients and what the industry is doing about them.
Okay, thank you, Justin. So we are now progressing to the next part here, impact of excipients in nitrosamine formation for drug products. So let's go first a little bit into the outline. So a few words will follow towards IFF, then about the impact of excipients and how IFF supports our customers, and then something new about the low nitrite excipient offering and future plans, if you want. So now about IFF Pharma Solutions. So we have decades of experience manufacturing excipients. Some of the names are here. And yeah, the most prominent is Metocell Polyox. So 10 manufacturing sites and 5 application centers across the globe. And we're committed to quality, manufacturing excellence, and provide our expertise in regulatory and polymer science. So next one. So how do excipients play a role in this nitrosamine discussion? So we received questions, especially at the beginning, whether our excipients contain nitrosamines. There was testing ongoing, and that was not the case, and it's very unlikely that it happens. There's in fact only one excipient that was found to carry nitrosamine. I think it was trolamine, so not one of our products. So that's very, very unlikely to be a contributor to nitrosamines itself. Then maybe some excipients contain vulnerable amines, but that's a pretty low probability. What is there in some cases, and it is nitrite, which is kind of ubiquitous in the environment, and so that was the key of the assessment that we have done. So sources of nitrite in excipients, where do they come from? So sometimes processed water can be a source. Even if it's purified, there's some low levels still of nitrite in there. Some of the raw materials are of natural origin, and they carry some trace levels of nitrites. And then in some cases can result from process impurity or reaction byproduct, depending on the route here. So zero nitrite, especially with the progress of the analytical testing, is something that is very unlikely to be achieved, at least in a very short notice. So then it was already mentioned that there was an industry initiative to monitor nitrite levels in excipients. It's the so-called LASA database, which is published and discussed in the literature. So the outcome, without looking into specific excipients, was that there's a wide variety of levels depending on the excipient, depending on manufacturers. And so the first thing was to find some transparency here regarding the levels. So the levels can vary, and nitrite is there in excipients. I think that's the line here. And so that was kind of call to action first for excipient manufacturers to test and assess the level of nitrite in the excipients. And there's a long list, if you remember the slide at the beginning, of excipients to be tested. So that was the first challenge. And then the other challenge was to find the right method, right, for each of the excipients. So, and we have some knowledge to share. We have a lot of excipients analyzed and developed methods for it. So sometimes, for example, you have viscous excipients. So if you try to extract them, you have difficulties, and then they can cause, in some cases, column fouling and degradation. So that can be avoided under certain circumstances. Then is the excipient soluble? So to freeze the nitrite, to analyze it, you need to know what solvents will work and not in the extraction. And then in some cases, methods were developed to avoid co-eluting peaks. So they could be there, like small molecules like acids, which co-elute with nitrite. So that could lead to overestimation. And so methods have been developed to address this point. So, but we're ready to have this discussion and help with the methods and direct customers to nitrite testing if they want to do it in-house. So this is about, let's say, our journey a little bit. So at the very beginning, we provided nitrosamine statements, make a big effort in analytical testing to have those statements for each product. So we're technically, an excipient supplier is not supposed to do so. Then we use the IPEC questionnaire, which is available for our customers already. And so it's a very structured template, which has all the information in there. And so if customers, for example, they have hundreds of excipients, and if you want to look up the nitrite level, you just go to a certain section of those and you find it there in a standardized format. So that has been found very useful by our customers. And then work was initiated very early to reduce nitrites and help our customers to manage then their nitrosamine risks, which is kind of something where the burden is with the marketing authorization holder, right? But we support our customers in their journey towards managing those risks. So one further here. So here you have kind of a setup. We typically use ion chromatography to analyze nitrite in our excipients. We developed validated methods for each chemistry and tested retained samples across the shelf life, more or less, and reported that values in our statement. As mentioned, these statements are available to our customers and have been found very useful based on the IPEC template in risk assessments for our customers. So one word to analytical methods. So there is different methods available. So some are based on reactions like the grease test. And we found ion chromatography more useful and even developed an ICMS method, so with mass spectroscopy detection, which is very specific for nitrite and can eliminate any potential issues with coelution, which are sometimes found in some of our excipients. So, and then let's say the outcome of the testing and some investment in the plans, we found or we developed low nitrite grades for Evisel, if you want. So there's certain grades which are available right now. So you see here a list of 5, and the nitrite analyzed so far is less than 29 ppb. So we're not talking ppms, but we're talking ppbs here. Those materials carry a specification, and the analytical method is designed to eliminate any coelution issues. The performance of those grades is equivalent to any other Evisel, as it's known for decades. We have addressed the regulatory dossiers as well, and samples are available. So that's a very good option to incorporate in the in any case that customers find a nitrosamine issue, and nitrite is a contributor. So word here to the testing and equivalence. So there's application testing that we have done, and we found that here, for example, the pH 102 and the pH 102 LN here in the compaction profile. There's a lot of other testing which is reported and has been conducted in all respects. Those products have been found equivalent. So there's one view into the specification sheet. So the specification is here. It's very small on my side, but you see here there's a nitrite specification which we provide on the COA as well, and each lot is analyzed and has a nitrite value on the COA. So that's very convenient to show control about nitrite. And not only about the microcrystalline cellulose, we as well looked into our hypromellose, the metocell, in the first step published the values in the white paper, which is available on request. So you see here the cake chemistries. We're very transparent here in the number of lots tested and the level found. So here it's in the ppb range. In some lots even have been found to be below the LOD of 8 ppb in this case. So then, before we enter Q&A, a little summary. So excipients should be considered in a holistic approach. So in case low nitrite is something that is needed, right? So then the excipients are there, but it's only one part of the holistic approach, and other contributors can play a role as well. The nitrite content can vary. Yes, that's true. And in the best case, customers receive a certification about this specific lot, what nitrite level is there. IFF continues to support and has been doing so since years, since 2 years actually, to support the risk assessment of customers and the product with discussions, with statements, and so forth. We have solid testing behind those statements. And then it's as part of the next phase, we have launched the LN for Evisel microcrystalline cellulose in response to customer needs. Where they're facing ever lower nitrosamine limits, right? And then as a further step, we're developing a low nitrite option for our metocell hydroxypropyl methylcellulose offering. So that's the end of my part, and I hope we have good time for Q&A, which will be moderated by Terry.
Thank you, Justin and Meinhof, for such an informative presentation. And we'll get started on the question and answer session now. I'd like to remind our audience how to submit questions. You can submit them by typing them in the Q&A box, which can be found directly below the video player. Now let's get to our first question. Does IFF offer low nitrite options for all of their excipients?
As it's obviously for me, right? So yeah, we're working on that as per customer demand. As mentioned, it's not so easy for each excipient to turn off the nitrite source. But if there's the demand, we're ready to take this up and work on that, right? So it's certainly not the end with Avicel and Metocel, right? So we're looking into more of the excipients as per customer demand.
Will procurement of excipients with low nitrites or nitrites on COA take a drug product view going forward, since not all APIs are susceptible high risks of nitrosamines, or will companies source according to worst case scenario? Do you need me to repeat that one?
Yes, please.
Will procurement of excipients with low nitrites or nitrites on COA take a drug product view going forward, since not all APIs are susceptible high risks of nitrosamines, or will companies source according to worst case scenario?
That depends on the company. So some prefer like an overall safety approach, right? But generally, it should be limited on a risk assessment. So the procurement of such low nitrite excipient should be based on a risk assessment. At least there should be secondary amine. Otherwise, it doesn't make sense. So, but it depends on the company, right? So we're ready to take the demand and see how we can serve.
Okay, thank you. Are IFF's nitrite testing methods fully validated?
Yes, that's a short answer.
Okay, let's go. Let's see. What's another one here? Is there any difference in specification between Avicel 102 and Avicel 102LN apart from nitrites?
No. Short answer as well. So the specifications are the same. Only the LN has an analysis of nitrite and a specification in on the COA. Yeah.
Thank you. Let's see. Next question here. What types of control strategies have been successful in minimizing nitrosamine formation? For example, using scavengers in the formulation, packaging options, etc.
I can take that one, Terry. Good
you.
question. I think the long and short is there's various and lots of different control strategies that could be effective. And it's going to be very dependent on the risk factors at play for that product. Now, I will say that, since it's mentioned in the question, there's several papers on the effective use of scavengers in a formulation. So if your product, if you cannot eliminate, the first and foremost, most effective control strategy is eliminating either the source of nitrosating agent, which Meinhof has really outlined a great way to do, minimizing nitrite impurities in your excipients. If that can't be done or done to an effective level, and the source of the amine cannot be eliminated, classic example is it's in the API structure, and that's there for a good reason other than nitrosamines. Then you look at things like scavengers, which have been shown to be very effective at eliminating or minimizing heavily nitrosamine formation. The other thing you can look at, particularly if you have a product in development, is where is the reaction occurring? So, for example, if I was developing a new product and I could not eliminate these sources, I would, in early formulation development, I would sample along the course of the formulation, right? If you're blending, sample the blend. If it goes into a granulator, sample after that. After drying, there's hypotheses that the actual act of drying a wet granulation can enrich formation because you're eliminating the water and concentrating the nitrite in solution. And I would test where the problem is and then determine, can you eliminate that problem? Can you mitigate it by shortening the drying time or minimizing the degree of mixing? Or if it's compression force related, can you lower the force, or can you deliver a blend? And those would be, approaches you would take if you can't use a scavenger because of compatibility reasons, for example, or you have a low risk and you're just trying to take some extra steps to mitigate it as a control strategy moving forward, to test those out and see if they're effective. So those are some examples of ways to do that. I know there's research, more broad research going out to say, if, for example, for API particles, can you coat the particle, almost putting either a chemical deposition on the surface of the API to basically separate it and isolate it from the excipients, and then upon ingestion with a coat would dissolve and release the drug. So these are some more advanced control strategies that are being looked at from a very academic research stance that might start to propose solutions going forward. But just wanted to kind of share that for folks to look into deeper if they have a problem that they can't get around.
Thanks, Justin. There's another question here that might be for you. The theoretical kinetic model was experimentally validated by the authors and demonstrated that the observed quantities were smaller. So would it be better to use experimentally observed conversion values in risk analyses?
Yeah, so if the question is around their particular product, I think to the degree that you can test your specific product to see if and where nitrosamines are forming, similar to the development example I just gave, absolutely go and do that. And the experimental data is going to prove a lot more valuable than the model, because certainly we know models are wrong. All models are wrong, and these models are very wrong. I think I articulated that. Now, what I should say is not appropriate is to use the experimental data in that paper directly to quantify your own case. And the reason for that is it's a different amine, it's a different solution. They're very specific set of conditions in terms of nitrite level in the solution and pH that may or may not be translatable directly to your product. So those are the considerations you'd want to take before leveraging that data from the paper and using it in your case. So hopefully I gave some sense of two different possibilities that could be behind that question. If you can characterize your own product, that's the strongest evidence you're going to get to being able to understand where the problem is and how to control it. If you can't do that, which often comes up in many examples, then leveraging the model is useful because it was very conservatively constructed. And I think, hopefully I articulated that in the papers going a lot of extent. So by that conservative nature, you can stand behind your predictions and justify them as being, again, conservative in worst case. And again, if your prediction is still well below the acceptable daily intake, you should have a strong argument why you have a no-risk scenario using the model itself.
Justin, is the quantification model for nitrosamine somewhere available?
Yeah, in the slide where I went over the construction of the model, there's a publication there. In fact, even the coding of the model is provided as the appendix or in a supplemental information. So it is available. You just need to construct it within your own software of choice, and build a user interface. I'm not aware, and it could be out there, I just might not be aware of someone that has put it in an available software that you can just buy off the shelf and start using.
Could the coating process increase the NNO amount, heat and moisture added?
Did this question just come in, or is this there for a while, Terry?
No, this was there for a while. Okay,
good. Yeah, I mean, the coating, I'm assuming this might be referring to like a tablet coating process. I'm going to go with that assumption. Certainly, in fact, we took a look particularly at tablet coating operations and found them to be negligible risk. And the reason for that is one is very, very limited surface to volume, right? You're coating large surface of a tablet so that the entire inside of that tablet is not getting direct contact with the moisture. The moisture in a coating process is evaporated very quickly. So you're getting limited absorption of that moisture to the tablet. And also the coating materials themselves are going to potentially have some nitrites, but again, on the surface of the tablet. So if your API or other amines is distributed throughout the tablet, you're only going to have contact for the surface. And as you start to build that coat, you're eliminating any other potential moisture excipients coming in from the additional layers of coat that you're putting on there from interacting directly with your tablet. So we've generally assessed, and it is present in the EFPA nitrosamine workflow, that coating operations are of low risk. The heat and moisture certainly are factors that can influence and enhance nitrosamine formation, right? But because of those other elements I articulated for the coating process, it wasn't deemed to be a very high risk operation in most cases.
Thank you. Here's another question. Nitrosamine risk assessment in API or drug product. Which approach is best?
Both are needed. So, early on, we felt, or the industry was thinking, that this was really only an API process or problem. And the reason for that is we thought drug products, particularly solid dosage products, were not going to be a high risk for nitrosamine or even see any formation. And the data that quickly rolled out showed that that's not the case. And in solid dosage products, you can have nitrosamine or even concerning level of nitrosamine formation. So the last time I worked regularly in this field a couple of years back, right, the guidance was specific to assess the drug product risk. If you want to do a drug substance risk assessment as well, you could, but you could not just only assess the drug substance. You had to assess the final drug product in the final packaging for the shelf life of the product in your risk assessment. So again, they both serve a purpose. If the only source of concerning or vulnerable amine that could react to form a nitrosamine is present in the drug substance, and you can control and end it there, then you can put the majority of your focus around the drug substance risk assessment. And if you show it's no risk there, and there's no additional risk that could carry into the drug product, and that's clean, then the drug product risk assessment can be pretty straightforward and easy, but it's still necessary to do according to the requirements by the health authority. So you have to do the drug product.
Okay, next question. Why nitrates aren't considered anymore on the nitrosamine risk assessment?
Yeah, that's probably for me. So the nitrates are considered like a negligible risk. So, and they have been removed from the IPAC template accordingly. So that's it. We received some questions about the nitrates. I don't know really where it comes from. It has no real base in literature. At least we haven't found that, and that has resulted in the IPAC template elimination of that. So the focus is on nitrite.
Well, I think we're out of time. There are a lot of questions that we didn't get to, but IFF will have access to your questions and can answer offline. And so with that, we're going to wrap it up. I want to thank the audience for attending and for participating in today's event. I'd also like to thank our sponsor, IFF Pharma Solutions, for making today's educational webcast possible. We'd like to ask everyone in the audience to participate in a brief survey. The survey will appear on your screen after today's presentation is ended. You'll receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who might have missed today's event. And with that, I want to thank you all for joining us, and we'll see you next time. Goodbye.